You're trying to watch a tiny molecular machine, way smaller than you can imagine, actually moving DNA.
Not just seeing it sit there.
Exactly, seeing it do the work. And when you finally get that glimpse, you see it uses this totally unexpected rippling wave motion, almost like, I don't know, folding DNA origami.
Right, it's pretty wild to think about.
It really is. And
Yeah.
getting this view, this like super detailed molecular dance, it's completely changing how we think about basic life processes and even huge things like cancer.
Mm-hmm.
So that's what this deep dive is all about today. We're jumping into chromatin, remodeling that absolutely essential process where proteins, kind of like tiny engineers, move the DNA packaging.
Nucleosomes.
Right, the nucleosomes around on the DNA. And this is crucial, right, because it controls if and when your genes get read.
Absolutely fundamental.
Our focus is on one key player, ISWI. It's an enzyme known for being really meticulous, setting at this precise, organized spacing between those nucleosomes.
Yeah, it's known for that regularity.
And our mission today, to unpack this honestly groundbreaking science paper that finally caught ISWI right in the act of remodeling. It's revealed secrets about how it works that, eluded scientists for years.
And connects it straight to human disease.
Exactly. So let's get into it. But first, maybe a little background.
Yeah, good idea. To really get why this is such a big deal, you need a bit of context. So our DNA, it's not just, a loose, messy string in the nucleus.
Right.
It's incredibly long. We're talking over 6 feet packed into a microscopic space, so it has to be tightly organized.
How does it do that?
It does this by wrapping around these protein spools called histones. 8 histones form a core, and the DNA wraps around it almost twice.
Okay.
And that unit DNA wrapped around the histone core, that's a nucleosome. And strings of these nucleosomes linked by little bits of DNA, that's what we call chromatin.
Like thread on millions of tiny spools, basically. But you can't read the thread if it's wound up super tight, can you?
Exactly right. If a gene is locked up tight in a nucleosome, the machinery that needs to read it, like for making proteins, often just can't get access. So the cell needs ways to, move those nucleosomes out of the way, and that's where chromatin remodelers come in.
The molecular engineers.
Yeah, they're enzymes that use energy from ATP, the cell's energy currency, to slide these nucleosomes along the DNA, or sometimes even change their structure.
Okay.
And the ISWI family specifically, they're important because they have this job of creating really uniform spacing, almost like perfectly aligned bricks.
That regular spacing sounds important.
Oh, it's crucial for controlling gene access, for maintaining the whole genome's organization.
So we knew ISWI existed, we knew it used ATP energy, and its job was this neat spacing. But you're saying we didn't really know the nitty-gritty, how it actually did the sliding.
Precisely. For years, we had models, sure, but they were based on indirect evidence. People proposed things like a step-by-step push, maybe a 2-step kind of thing.
Inferences, basically.
Yeah, exactly. Inferences from seeing what happened before and after. We didn't have that real-time picture of the middle steps, how ATP energy actually turns into physical DNA movement around the histone spool.
And that missing piece wasn't just curiosity, right? It matters for disease.
Absolutely. Understanding this basic action and how it can go wrong is key for diseases like cancer, where chromatin organization often goes haywire, and also for many developmental disorders linked to these remodelers.
Okay, so the big challenge, catching something microscopic moving incredibly fast right in the middle of its job. How did these researchers finally manage it? How did they get that look at ISWI while it was sliding the nucleosome?
Right. This is where the tech gets really exciting. Their main weapon was time-resolved cryo-electron microscopy, cryo-EM.
Okay, cryo-EM, taking super high-res pictures of frozen molecules.
Pretty much. You flash freeze your sample super fast in thin ice, then shoot electrons through it to get an image. You can see structures at almost atomic level detail. But the breakthrough here is the time-resolved part.
So not just one picture.
Nope. A series of pictures at incredibly short time intervals.
Like a movie camera, but one that can see atoms.
That's a great way to put it. And the resolution they got was just astonishing. Down to 2.3 angstroms.
Wow. Remind me how small an angstrom is.
Well, think of a human hair maybe 800,000 angstroms wide. A carbon atom is roughly 1 angstrom. So 2.3 is getting close to seeing individual atoms' chemical bonds.
That's incredible resolution.
Yeah.
But okay, how do they make sure they froze it at just the right millisecond? If it's happening that fast, how do you catch those fleeting moments?
That's where they got really clever. It was a mix of chemistry and microscopy. The key was synchronization.
Getting everything to start at the same time.
Exactly. Instead of just mixing everything ISWI, nucleosomes, normal ATP, and, hoping for the best, they used a specially engineered ATP. It's called photoactivatable caged ATP.
Caged ATP, like it's locked up.
Kind of. It's like normal ATP, but it has this chemical cage on it, making it inactive. But here's the trick. You can break that cage open instantly with a pulse of UV light.
Ah, so they set everything up, ISWI, nucleosome, the caged ATP let it settle, then flash. Start the reaction for millions of molecules all at once.
You got it. That synchronizes the starting gun.
Yes, yeah.
Then they use this rapid sample delivery system to spray the reacting mixture onto the cryo-EM grids and freeze them at specific, really short time points after that flash.
Like 5 milliseconds, 25 milliseconds, snapshots in time.
Exactly. Capturing the enzyme in different unstable transient states during that ATP cycle and the remodeling.
Okay, so with these atomic level, time-stamped snapshots, what did they actually see? Was ISWI just like shoving the nucleosome along?
Well, this is where the big surprise came in. They did see a pushing motion from a part of the enzyme called the HAND-SANT-SLIDE domain. That's the motor part.
Okay.
It grabs the DNA and moves forward, pushing about. Look at this. 2 base pairs of DNA towards the 3-foot end.
Only 2 base pairs. That seems really small. If you're trying to move a whole nucleosome with nearly 150 base pairs wrapped around it.
It is small. And that's exactly what tipped them off to their really novel model, this origami DNA wave.
DNA wave. Okay, explain that.
They realized the overall movement isn't one big shove. Instead, the sliding happens through this much more localized dynamic thing where the DNA itself gets distorted.
The enzyme folds the DNA.
Sort of. It induces a small temporary fold or bulge in the DNA path around the nucleosome, specifically near a point called SHL2 where the DNA bends sharply anyway. This distortion is like a tiny wave or a ripple that moves along the DNA track relative to the histones. And the remodeling happens because this microfolding and unfolding repeats maybe every 10 base pairs or so.
A DNA wave.
Yeah.
That's really counterintuitive. We think of DNA as, this is a stable helix that sounds way more elegant than just pushing. Did they confirm this wave idea somehow, besides the pictures?
Oh, yeah. They didn't just rely on the structures. They combined it with another technique. High-throughput single-molecule FRET.
FRET.
Yeah.
That's where you put fluorescent tags on things and measure distances.
Exactly. First-order resonance energy transfer. You attach these little glowing labels to specific spots on the DNA or the enzyme. Then you measure energy transfer between them, which tells you about distance changes and movements in real time for single molecules.
So they could actually watch the DNA points moving relative to each other, like see the wave happening dynamically.
Precisely. And this gave them the quantitative data to back up the cryo-EM snapshots. Using FRET, they measured the speed of this DNA distortion, this wave, about 0.8 base pairs per millisecond.
Wow, so the techniques really complemented each other.
Beautifully. The FRET added the motion and speed to the structural detail from cryo-EM.
Okay, so we have the wave, but you mentioned they captured lots of different states, right? Not just the wave moment.
Absolutely. Freezing at multiple time points gave them like 9 different high-resolution structures. It's basically a molecular movie of the ISWI cycle.
What kind of states?
They saw the enzyme bound to ATP, bound to ADP, that's the used ATP, and this crucial ADP state where they actually saw the DNA bulge, that 1 base pair distortion, plus states with no nucleotide bound at all.
So different frames in the movie.
Exactly, showing how the enzyme changes shape as it burns that ATP fuel.
And seeing all those different frames.
Yeah.
What did that reveal about how ATP energy actually drives this specific wave-like motion?
By comparing the structures, they could pinpoint the key parts inside the enzyme. They found specific bits, motifs V and psych, that act like little relays or linkages.
Relay?
Yeah, they transmit the shape changes happening where the ATP binds, when the energy is released,
Right.
all the way over to the parts of the enzyme gripping the DNA.
Connecting the fuel to the action.
Right, and that signal transmission causes that critical 1 base pair DNA bulge, especially in that ADP state. That seems to be the power stroke.
Okay.
They also saw how the movement of the DNA strand that the enzyme holds onto, they call it the tracking strand, makes it temporarily break contact with the histone core at a specific spot, SHL 2.5.
So it's this whole coordinated dance. Shape changes, signals, DNA distortion, letting go of the histone for a moment.
Exactly, all allowing that wave to propagate along.
So they've pretty much cracked the code on the engine itself, how it uses ATP for this wave motion.
Hmm.
But like you said, biology needs control. You don't want ISWI just surfing DNA waves nonstop. Is there a brake pedal?
That's another really critical piece they uncovered, a regulatory layer. They found a specific mechanism that acts, like a brake on ISWI's activity.
A brake? How does a tiny protein machine hit the brakes?
It's structural. They found this feature, a helix structure that forms transiently. They called it the brake helix.
Okay.
This helix forms specifically when the enzyme is in a certain state, like the ADPB state, which seems to be kind of an off-ramp from the active cycle.
And what does the brake helix do?
When it forms, it interacts with another part, the gating helix. And this interaction forces the whole enzyme into this weirdly open, inhibited shape, like locking it in off.
Clever. Why is that brake so important?
Well, it seems to help ISWI sense the local environment, particularly the length of the linker DNA that's stretched between nucleosomes. ISWI needs to create regular spacing, remember?
Right.
This break helps it stop if the spacing is already right, or maybe if the linker DNA is too short. It prevents it from, messing things up by remodeling too much or in the wrong place.
Okay, this is amazing. We've gone from just sliding to this detailed ratchet and wave engine with a built-in environment sensing brake. What does this huge leap mean for science more broadly?
Well, first, obviously, it gives us that quantitative molecular model for ISWI we were missing. It explains how it creates that regular spacing. It's a big theoretical jump.
Beyond just ISWI, though.
Oh, definitely. The technical side is huge, too. They basically pioneered this robust way to do cryo-EM on a millisecond timescale.
So they didn't just answer their question. They built a better tool for answering lots of questions.
You got it. This whole approach, synchronized reactions with those photocaged molecules, plus rapid freezing for cryo-EM, it's a powerful new tool for structural biology.
What else could it be used for?
Think about any complex molecular machine that acts fast. DNA transcription, replication, protein synthesis, transport. Anything involving quick shape changes or movements. This technique could potentially shed light on all sorts of dynamic processes.
Wow. Okay, and let's circle back to cancer. You mentioned the connection. How does this detailed mechanism link back to disease?
Right. This is where it gets very direct. The researchers looked at data from human cancers. They analyzed genetic data from over 8,000 tumor samples across different cancer types.
And they looked for ISWI mutations.
Yep. And they found specific common mutations. K588E and K593N were two big ones located smack dab in the region. Their structures identified as the core ratchet axis.
The very heart of the wave-making engine.
Exactly. The part involved in turning ATP energy into that DNA movement.
So these common cancer mutations hit the engine directly. What happens then?
They tested it. They put those specific mutations into ISWI in their experiments, and they found these mutations significantly slashed the remodeling rate, cut its activity by about half. So by understanding the precise mechanism, the ratchet, the wave, the energy use, they've shown how these cancer mutations break ISWI. They mess up that specific motion needed for proper nucleosome spacing.
Which leads to messed up chromatin.
Which is a hallmark of cancer cells. It gives a clear molecular explanation for how these mutations drive the disease.
That is incredibly powerful. Linking atomic detail and millisecond timing to the cause of a disease. Does knowing it at this level suggest new ways to treat it?
It absolutely does. When you know the exact molecular surfaces involved in the stepping, the wave, and especially that regulatory break,
you get targets.
Highly specific targets for drug development. For cancers where ISWI is mutated or just acting up, you can now think about designing drugs to specifically block those interactions, maybe inhibit that aberrant remodeling, targeted therapies.
So this deep dive started with trying to see a tiny machine move, and it's given us this incredible view. ISWI's dance with DNA, the surprise DNA wave, the regulatory break, and a direct link to cancer mutations found in thousands of patients. Quite the journey. Are there still big questions left? Next steps.
For sure. While this study is amazing for the core mechanism, it mostly used a pretty simple system, right? A single nucleosome, maybe two.
Real chromatin is messier.
Much messier. It's packed into fibers, interacts with tons of other proteins, things like transcription are happening nearby. So a key next step is verifying if this exact wave mechanism and the break work the same way in those more crowded physiological settings inside a living cell, ideally.
Makes sense.
But the core mechanism they've uncovered and the technical leap they made, that's incredibly impactful. It really sets the stage for future work.
So you've taken a deep dive into how the ISWI chromatin remodeler really works, going way beyond old models to see this intricate dance, complete with that surprise DNA wave and a clever break.
It really does reshape how we picture these essential machines organizing our genome. It highlights the, I don't know, the elegance and complexity hidden inside our cells.
And it's a powerful reminder of how getting down to these molecular details using cutting-edge techniques like time-resolved cryo-EM can directly light up the causes of diseases like cancer.
And point towards new ways to tackle them.
Definitely. So here's something for you to mull over. If ISWI uses this unique wave to move DNA, could similar sort of non-intuitive wave-like mechanisms be happening in other really fundamental DNA processes?
Interesting thought, like in DNA replication or repair.
Maybe, with totally different machines.
Yeah.
And if we could understand and maybe precisely control that specific wave motion, turn it up, turn it down.
Yeah.
What could that mean? For disease or even for manipulating how cells work?
Lots to think about there.
Definitely something to chew on until our next deep dive.